Hey guys, hope you are doing well. It's Rashid Hashmi from Doctors Digital College. Here you will get video lectures for O and A levels of Cambridge Assessment International Examination, IGCSE, GCE and Edexcel boards. We'll also include lectures for f.sc, metric and anti-test for different subjects. Hope you will enjoy it.
Welcome to Doctors Digital College.
In this lesson, we will discuss about oxidation of pyruvate, Krebs cycle and oxidative phosphorylation, which is also called electron transport chain. So let's see. In respiration, after glycolysis, the next step which we have devised for our easy to study is link reaction, which is also called oxidation of pyruvate. The product of glycolysis, as we have already studied, is pyruvate, along with some other products of energy. Pyruvate is produced. Pyruvate is a 3-carbon compound, and now pyruvate is moved from cytoplasm into mitochondria. From cytoplasm to mitochondria it will be moving, and here there is the use of ATP energy for the movement of pyruvate from cytoplasm to mitochondria. This is one of the reasons why 38 ATP moles are not produced by the oxidation of 1 mole of glucose, because energy in the form of ATP is being consumed for such type of transports. Now pyruvate is changed into acetyl coenzyme A. Pyruvate is a 3-carbon compound, while acetyl coenzyme A is a 2-carbon compound. It means along with acetyl coenzyme A formation, there is the formation of carbon dioxide. 1 carbon is released, which is changed into carbon dioxide. You know that the group of acetyl is like CH3C double bond O, like this. This is acetyl. And here along with it, there is coenzyme A is attached like this, S-CO-A. This is the part of acetyl coenzyme A. In some next lecture, we will study that what is the role of acetyl coenzyme A, what is the role of NAD, and what is the role of FAD, which is going to be used in Krebs cycle. Now 3-carbon compound has been changed into 2-carbon compounds. There is the removal of carbon. So whenever there is the removal of carbon, there is the removal of hydrogen also. In case of carbohydrates, every carbon is having hydrogen attached with it. So along with the removal of carbon, there is the removal of hydrogen also. And that hydrogen is taken away by NAD, which is reduced into NADH2. For easy to study, I am once again writing here NADH2. What does it mean that NAD is being reduced and pyruvate is being oxidized? So this is the oxidation of pyruvate into acetyl coenzyme A, whereby the oxidizing agent is NAD, which is itself reduced into NADH2. If we try to balance this equation, from glycolysis we got 2 pyruvate. Okay, so here we should multiply that we have 2 moles of pyruvate from 1 mole of glucose, and we are getting here 2 moles of acetyl coenzyme A and 2 moles of carbon dioxide. Now this acetyl coenzyme A is present in mitochondria and is being attached here like this. Coenzyme A is attached here. So this is the overall reaction I have written here. There are some intermediate steps, but during those steps there is no energy change or no carbon change. The energy by this oxidation process has been stored in NADH2 in the reduced form of NAD, which will be given out in the next stage. Now this stage in the mitochondria will occur only if oxygen is available. Or if oxygen deficiency is there, then some of the pyruvate will not be entering into the mitochondria, rather it will go for anaerobic respiration. And in the complete absence of oxygen, such as in case of some microorganisms or some roots of the plants, their pyruvate will not enter into mitochondria, means it will not enter into mitochondria at all, and all of it will be going towards anaerobic respiration, that is called alcoholic fermentation. We will discuss about alcoholic fermentation and lactate fermentation in some next lecture. So this is the oxidation of pyruvate, and the next step is Krebs cycle. Krebs cycle is also occurring in the mitochondria, and we should write here specifically that these two steps are occurring in the matrix of mitochondria. Link reaction and Krebs cycle, they are occurring in the matrix of mitochondria, whereby the next step, which is oxidative phosphorylation or electron transport chain, that is also occurring in mitochondria but on the surface of inner membrane of mitochondria. Now let's see what is happening during Krebs cycle. If I write here the overall process of Krebs cycle, then it is like this, that it is here acetyl coenzyme A, which is a 2-carbon compound. It is entering into a cyclic process like this, and during this cyclic process there is the removal of these 2 carbons in the form of carbon dioxide. It is here carbon dioxide and it is here carbon dioxide. Like oxidation of pyruvate, along with the carbon dioxide formation, there is the formation of NADH2, NADH2. And along with it, apart from these two NADH2 formation, there is another NADH2 produced here, and there is one FADH2 produced. And all these compounds, NADH2, FADH2, they are having energy stored with them. Along with it, there is also substrate level phosphorylation, whereby there is the formation of ATP. Adenosine triphosphate is also formed through the formation of GTP. We will study in detail that what is happening in overall this process, that these 2 carbons of acetyl coenzyme A are given out in the form of carbon dioxide. Now once again, if I try to balance this equation, from glycolysis we got 2 pyruvate, and from 2 pyruvate we got 2 acetyl coenzyme A, and if I write here that we are having 2 acetyl coenzyme A, means total we have 4 carbon, then we should multiply the whole of this cycle with 2, means we are getting 2 carbon dioxide here, 2 carbon dioxide here, 2 NADH2, 2 NADH2, 2 FADH2, 2 NADH2. So as a result, we have got out of this Krebs cycle, if I write here like this, that I have got, what is the product? The product is 2 carbon dioxide plus 6 NADH2, rather 4 carbon dioxide, 6 NADH2 plus 2 FADH2 plus 2 ATP. This is the overall product of this Krebs cycle, if we get the balance of this whole equation. Now let's see what is happening in this process. What are different biochemical processes occurring during which carbon dioxide is given out and energy is given out. Now let's see what's happening in Krebs cycle. From link reaction or the oxidation of pyruvate, we got acetyl coenzyme A. Acetyl coenzyme A is a 2-carbon compound. With acetyl, a carrier coenzyme A is attached. Now it will enter into a cyclic process which is called Krebs cycle. Krebs cycle is occurring in the matrix of mitochondria. Now the stage it will enter into this cyclic process, the last product here is oxaloacetic acid, which is a 4-carbon compound. Now this oxaloacetic acid will take this acetyl coenzyme A and is changed into citric acid. Citric acid is a 6-carbon compound. This citric acid is having 3 carboxylic groups. C double bonded O, OH. 3 carboxylic acids are attached. So this citric acid is also called tricarboxylic acid. Or TCA. Tricarboxylic acid or TCA. Because of this first product formed after the entry of acetyl coenzyme A, Krebs cycle is sometimes referred as citric acid cycle, or it's also called TCA cycle. Now, let's see what's happening in this process. That citric acid is first of all changed into a 5-carbon compound called alpha ketoglutarate. This is a 5-carbon compound. Glutaric acid or glutarate. What does it mean? That 1 carbon is being removed in the form of carbon dioxide. Once again, I am omitting some of the intermediate steps during which there is no carbon change or during which there is no energy change. If carbon is being removed, it means hydrogen is also being removed, just like that in link reaction. And the removal of hydrogen here is called oxidation. So citric acid is being oxidized into alpha ketoglutarate. Okay, if citric acid is being oxidized, there must be some oxidizing agent. The oxidizing agent here, it is NAD, which itself is reduced into NADH2 during this process. Okay, now in the next step, alpha ketoglutarate is oxidized into succinate, which is a 4-carbon compound. Once again, during this process, 1 carbon is being removed, and along with the removal of carbon, there must be the removal of hydrogen, and that removal of hydrogen is by the oxidation of NAD. So in this stage, I am once again writing that NAD is reduced into NADH2, whereby there is the oxidation of alpha ketoglutarate into succinate, and there is the reduction of NAD into NADH2. Now, this oxidation process is more exergonic as compared to this oxidation process. So, what happens during this oxidation process? A lot of energy is stored, almost equal to 3 ATP, is stored in NAD, and along with it, there is some energy given out in the form of this. GDP is changed into GTP. Guanosine diphosphate is changed into guanosine triphosphate, which later on will be used for the conversion of ADP into ATP. So we can say along with the formation of NADH2, there is also the energy stored in the form of ATP. This is another substrate level ATP formation. Now, in the next step, succinic acid is changed into fumaric acid. Fumaric acid is 4-carbon compound, and fumaric acid is then changed into malic acid, and then malic acid is changed into oxaloacetic acid. Now you can see that oxaloacetic acid is a 4-carbon compound, succinic acid is a 4-carbon compound. It means after the succinate formation, in all the other steps there is no removal of carbon. Means these 2 carbons of acetyl coenzyme A, they have been removed here in the form of carbon dioxide and carbon dioxide. There is no removal of carbon. But there is oxidation, such as during the conversion of succinate into fumarate or fumaric acid, there is the oxidation process, and this oxidation process is, I should write it this way, that FAD is reduced into FADH2. Okay? Fumaric acid is then changed into its isomer malic acid, and then when malic acid is changed into oxaloacetic acid, there is once again NAD reduced into NADH2. Means once again malic acid is oxidized into oxaloacetic acid, so there is the reduction of NAD into NADH2. So overall in this process, if you look at this, as we have discussed already, that there are 1, 2 and 3 NADH2 formation, and there is 1 ATP formation. Okay? During these 2 processes, there is decarboxylation and dehydrogenation. As I have shown here in green, it means there is dehydrogenation along with decarboxylation. This is oxidation process. But as I have shown here in red, it is only the oxidation process. There is no decarboxylation process. Now, one thing more in this case, I would like to tell that sometimes we need to write the name of some enzyme being used. Here, we are not using the specific name of enzymes. The name of enzymes are actually derived. How are they derived? That we will derive it, that we will use substrate, the name of the substrate, plus process, what is the process occurring, and plus is added. The word is added. Say for example, in this case, what is the enzyme being used? In this case, the substrate being used is citrate. Okay, and the process being done is decarboxylation. Decarboxylation process is occurring. So the name of enzyme will be, by joining all these 3, there will be citrate decarboxylase. Citrate decarboxylase is the enzyme being used here, which is removing the carbon. You know, there is also the removal of hydrogen at this stage. So there is another enzyme called citrate dehydrogenase used. So if we will write here, there will be alpha ketoglutarate decarboxylase, alpha ketoglutarate dehydrogenase. So is here we can write the name. So we are not using specific names, rather we can use this type of names. Now, once again we will balance this equation. From, if we are starting once again from glucose, during glycolysis we got 2 pyruvate. 2 pyruvate were changed into 2 acetyl coenzyme A by the link reaction or by the oxidation of pyruvate. And now in Krebs cycle, 2 acetyl coenzyme A will pass through this cycle, means the products of 1 mole of glucose will run Krebs cycle twice. So I should write that the whole of this process is multiplied by 2. Means there are 1, 2, 3, 6 NADH2 produced, 2 FADH2 produced, and 2 ATP produced. Now we have to see that what is happening to the NADH2, FADH2. Because I told that link reaction and the Krebs cycle, they will occur only if oxygen is available. Or if sufficient oxygen is available, then these processes will be occurring. If there is a deficiency of oxygen, then these processes, such as in case of mammalian muscles, these processes will occur up to the extent that oxygen is available to oxidize NADH2 into NAD. So in the next step, which is called oxidative phosphorylation, or which is also called electron transport chain, overall process being done is that NADH2 is oxidized into NAD. This process is occurring in the next step. Where are these two hydrogens going? These two hydrogens are being taken up by molecular oxygen. That it is here molecular oxygen entering and it is forming water. Now, what has happened to the energy which was stored with NADH2? That energy has been given out in the form of 3 ATP. 3 ATP are produced by the oxidation of 1 mole of NADH2. 3 moles of ATP are produced. And from 1 mole of FADH2, ideally 2 moles of ATP are produced. So we will see in the next step that how is this process occurring on the inner membrane of mitochondria. The next process is oxidative phosphorylation, or it is also called electron transport chain. Or in simple I can write it like ETC, or sometimes it's also called as oxidative phosphorylation. We also called it oxidative phosphorylation. What happens actually during glycolysis, link reaction and Krebs cycle, there was a lot of production of NADH2, FADH2, and there is a lot of energy stored in FADH2, NADH2, and energy present in all of them is given out. If this is here mitochondria, this is its inner membrane. We will study in detail what is the structure of mitochondria in relation to the cellular respiration. Here on this surface of the mitochondria, this inner surface of the mitochondria, this oxidative phosphorylation or electron transport chain is occurring. If I draw it here as large size, that this is the outer membrane as a bilayer and this is the inner membrane as a bilayer. This is here inner membrane of mitochondria, and on this inner membrane of mitochondria there are F1 particles present. There are large number of F1 particles present, and along with it there is also electron transport chain present on the inner surface, on this surface. Okay, here on this surface there is oxidative phosphorylation process occurring. Now let's see how is this process being done. If we start it with NADH2, when NADH2 is oxidized into NAD, there is the reduction of FAD into FADH2. Now there is the energy difference between NADH2 and FADH2, and we can see that some energy is given out at this stage. Actually there is no energy given out at this stage. This energy difference between NADH2 and FADH2 is being stored as electrochemical energy, which will be used at the last step. Now the next step is that FADH2 is oxidized into FAD, and hydrogen, electron and energy is given to a complex of enzymes called coenzyme Q. This complex of enzymes, coenzyme Q, the job of it is that it will separate protons from electrons. Electrons and protons, they are separated. What happens at this stage? Protons are sent into the space between the two membranes of mitochondria, while the electrons will be moving to the next. Now electrons are given to the electron transport chain. These electrons, they are going from one cytochrome system to another cytochrome system. Means electron transport chain is comprised of cytochrome system. Now the next step will be cytochrome B to cytochrome C to cytochrome A. Means there are many other cytochromes, but just for the short, I have written those cytochromes during which energy is given out. From cytochrome B to cytochrome C to cytochrome A, or cytochrome A3 and A1, electrons are moving. And when they are moving, energy is being given out like this. Means electrons are being lowered in their energy level. From their higher energy shells to lower energy shells, they are coming. But what's happening to this energy? Is there ATP production at this stage? No, ATP production is not there. What's happening? That protons and electrons, they are being separated from each other. And you know, what is this process done? Separation of positive and negative charges. This is just like the production of electricity. When turbine is running, what's doing? Turbine is separating positive and negative charges. If fuel is being burnt, engine is running, or if the waterfall is there, water is falling on it, potential energy of the water is running the turbine, and the turbine is separating positive and negative charges. And that is called electricity. And the electricity we are consuming at our houses or in different processes is actually the joining of positive and negative charges. When positive and negative charges will join, energy will be given out, and that energy is then converted into different other forms such as heat energy or light energy or kinetic energy or some other form of energy. So is here that chemical energy present in reduced form of NAD or in reduced form of FAD has been used up till now for the generation of electricity. Electrochemical energy has been produced. That is the separation of positive and negative charges. Now ultimately at the last step, these positive and negative charges, they will join with each other. They will join with each other on the head of stop particle, and their energy given out will be used, and that will be stored, you could say, in the form of chemical energy. And the chemical energy is ATP energy here. Okay, so let's see what's happening. These protons, they are coming out from the head of stop particle. These are here 2H positive. Say for example, I am writing here 2H positive, and the electrons are coming from here. These are the 2 electrons. Just for example, I am writing here the 2 electrons. Now these 2 electrons, they are taken up by molecular oxygen. I am writing here half the molecule of one oxygen, and it is changed into O-2. Now these two things, O-2 and 2H positive, they will join with each other to form H2O. Water is produced. Now what has happened? Water has been produced, but actually there is the joining of positive and negative charges. These protons and electrons, they have joined. Although they have attached with the chemicals and then they have joined to produce water, but there is the consumption of electricity. Electrochemical energy have been consumed. So where is that electrochemical energy going? That electrochemical energy is actually used here for the formation of bond between ADP and inorganic phosphate, which will be forming ATP. ATP formation is there. If we are starting from reduced form of NAD, 3 steps are shown here which are giving out energy. So electrochemical energy is the magnitude or the quantity of electrochemical energy is up to such an extent that 3 of such bonds can be formed. Means the bonds between 3 ADP, 3 inorganic phosphates can be formed to make 3 ATP. Or if we will be starting with FADH2, the first step will be omitted, and then the electrical energy produced here will be sufficient to make 2 of high energy phosphate bonds. Now, this process of joining of inorganic phosphate with ADP to produce ATP is called phosphorylation. As we have studied, during glycolysis there was phosphorylation, during Krebs cycle there was phosphorylation through GDP to GTP and then ADP to ATP, and that phosphorylation is called substrate level phosphorylation. And the phosphorylation process being done here, this is called oxidative phosphorylation, because this phosphorylation is occurring in the presence of oxygen. Molecular oxygen is there, the oxygen which we are inhaling through blood, it's going into the cells, and there inside the cells, inside the mitochondria, this is being consumed like this. It is being changed into water. So this is oxidative phosphorylation. Now, different other names are given to this process, which is also called oxidative phosphorylation. Sometimes there is a term used, chemiosmosis. What is this term, chemiosmosis? You might have studied that osmosis is the movement of water across some membrane. That when water is moving from higher water potential to the lower water potential through some membrane, it's called osmosis. If you look at this membrane, this is a membrane of the cell, this is a bilayer membrane, water is there on one side of the membrane. So, is this osmosis? But here is no water on the other side of the membrane. Now, on one side of the membrane water is present, on the other side of membrane water is not present. That's why we have devised a different term rather than to use osmosis. We have devised a different term called chemiosmosis. Means the osmosis through the chemicals. Chemicals are joining to make water. So here it's appearing perhaps water is moving through the membrane, but actually on the other side of the membrane there is no water present. So for oxidative phosphorylation, the process occurring at the head of stalk particle or the F1 particle, this term chemiosmosis is also used. There is another term called couple reaction. Couple reaction term is also used for this process. Couple mean two. The two processes are being done. What are those two processes? One is the phosphorylation and the other one is water formation. There is the formation of water and there is the formation of ATP. So these two processes, they are occurring side by side. That's why the overall biochemical process being done on the head of stalk particle or at the F1 particle is also called couple reaction. This F1 particle is protein in nature. It has a specific structure, and when protons are passing through it, it is revolving like this. Okay, so that the electrical energy is passing through it and it's moving, and that electrical energy is used here. I hope that this process of link reaction, Krebs cycle and electron transport chain or oxidative phosphorylation is well understood to you. And I hope that you will log on to my channel, you will subscribe my channel, or you will also log on to www.doctorsdigitalcollege.com for having similar lectures for other subjects also.